Good morning and welcome to XenoTech's webinar, Aldehyde Oxidase, Why In Vitro Absolute Activity Doesn't Matter. My name is Matt Beck and I'm the Marketing Manager here at XenoTech. Today I am pleased to be joined by members of the XenoTech Consulting Group, Mr. Brian Ogilvie and Faraz Kazmi. Before we get started with today's presentation, I would like to go over a few housekeeping items. Here's an example of your GoToWebinar user console. Using the orange arrow will allow you to minimize and maximize the user console. Please note that your audio is muted. If you have questions during the presentation, please post your questions in the questions pane seen highlighted here. We will have a Q&A session immediately following today's presentation to address questions posted via the questions pane. A copy of the slides and recorded session will be available a few days after the presentation. We will send both links via email for your reference. These links will also be available on the XenoTech website in the coming days. It is important to know that whatever email address that you've signed up with through GoToMeeting is the email address that we will be sending the presentation slides and recorded version to. So with that, I am pleased to have two members of the XenoTech Consulting Group with me today. I'm joined by Principal Scientist Mr. Brian Ogilvie and Senior Scientist Mr. Faraz Kazmi. Without further ado, I'm going to turn it over to Brian to get us started. Brian?
Okay, thanks Matt for the introduction. I'm going to switch over here to our slides, and again, as Matt mentioned, the title, Aldehyde Oxidase, Why In Vitro Absolute Activity Does Not Matter. And we'll go through, I want to orient people to the importance of aldehyde oxidase, and then go through how there's a variable activity between cytosol and S9 and possible reasons for that, as well as Faraz will then cover the impact of low and high aldehyde oxidase activity lots on in vitro to in vivo extrapolation, and then also go over the calculation of the fraction metabolized by P450 versus aldehyde oxidase. So again, I know a lot of people are familiar with aldehyde oxidase, especially in the last several years. It used to be sort of a metabolic curiosity. Now I think it's become more mainstream. But it is a molybdenum-containing enzyme that does not require NADPH. And there are others, including xanthine dehydrogenase, oxidoreductase rather, as well as the mitochondrial amidoxine-reducing components. But aldehyde oxidase is one that's present and does not require a cofactor and metabolizes a wide variety of drugs. So it also has relatively wide tissue distribution, so it's high in liver, kidney, and lung. And in humans, it has generally high but quite variable activity. It does have cytosolic localization, as I mentioned, and the wide substrate selectivity includes aromatic aldehydes as well as aromatic azoheterocycles and aminium ions that can actually be produced as intermediates from P450 metabolism. Reductions have also been reported, so with ziprasidone, some isoxazole moieties, nitroaromatics, N-oxides, and sulfoxides. And in some cases, you do have to perform these in vitro under special conditions, so their clinical relevance, with the exception of ziprasidone, is not widely known at this point. But the major point that we'll cover is that there are major species differences, and these can lead to erroneous prediction of human clearance. So for instance, dog, the aldehyde oxidase gene is a pseudogene. In rat, you have moderate but quite variable activity depending on the strain. Mouse, guinea pig, and rhesus monkey have generally been considered to have high aldehyde oxidase activity, but as you'll see, that's somewhat substrate dependent. So aldehyde oxidase metabolism in humans has actually led to the withdrawal from development of several drugs, including Pfizer's CP945863, carbazaran, and RO1. So to give you just a simplistic overview of drug design for low CYP liability, what most people have been doing for the last several years is reducing the overall log P, and that's generally because there's at least a loose correlation between high log P and CYP metabolism, but this is not true for aldehyde oxidase metabolism. There appears to be very little correlation in terms of the normal physical chemical properties of drugs and its propensity to metabolize. So one of the things you can do is add fluorine to certain sites on a molecule to block oxidation by P450, and then to just simply reduce the electron density of aromatic carbons. So for instance, introducing nitrogen, and because P450s tend to oxidize carbon atoms with high electron density, And aldehyde oxidase tends to oxidize carbons with low electron density. You're now switching to some extent from P450 to aldehyde oxidase. So Deepak Dalvi had a nice presentation at the Gordon Research Conference a couple of years ago where he presented these structures. So for instance, with naphthalene, you have only P450 metabolism. This intermediate structure, quinoline with one nitrogen, you have both P450 and aldehyde oxidase. And once you get then to quinoxylene with two nitrogens introduced, it's aldehyde oxidase only. But these are small molecules, and I will mention Deepak Dalvi and others have done some additional work with larger molecules where the spatial orientation and modifications in the binding pocket of aldehyde oxidase is also important. So these rules are not absolute, but they are general, allow you to look further into which compounds would be metabolized by aldehyde oxidase. So this again is from another review paper coming out of Pfizer where they have a list of model azoheterocycles that may be metabolized by aldehyde oxidase. So it's a nice graphic to look at when you're looking at the potential for your drug to be metabolized by aldehyde oxidase. If you have one of these moieties that's in an exposed area of the molecule, you might possibly see aldehyde oxidase metabolism. So to give you an idea of some of these other structures that we've used as model substrates here at XenoTech, Faraz will talk a little bit more about some of these. Our typical ones are these small molecules, so phthalazine and vanillin, some of the classic aldehyde oxidase substrates. But we've also then looked at, for instance, carbazeran, which has high intrinsic clearance by aldehyde oxidase, O6-benzoguanine, medium clearance drug, and zaniparide, medium intrinsic clearance drug, as well as a low intrinsic clearance drug, so methotrexate and zaleplon. And I also wanted to point out 6-deoxypencyclovir, and this is actually a prodrug which is actually activated by aldehyde oxidase. So aldehyde oxidase can obviously be very important for enzymes with high fraction metabolized by aldehyde oxidase. And because of this trend for a move away from P450 metabolism, it leads to more aldehyde oxidase liability, if you will. So higher clearance in human hepatocytes versus human liver microsomes, if you do those studies, can be a clue that you have aldehyde oxidase metabolism. So then you would want to look for these aromatic azoheterocycles and other AO structural motifs to see if that's the case. And as I mentioned, dramatic species differences for some AO substrates. So when you're in your preclinical species, the clearance may appear quite reasonable, and you would predict that you're going to have a decent half-life in humans. But then when you actually get to humans, and this has been described in various presentations and papers, you can have very high clearance in humans. One of the problems, as I mentioned, dogs completely lack aldehyde oxidase, and really even for the species that do express aldehyde oxidase, they express multiple forms, and even rhesus monkey or guinea pig cannot predict human clearance for every AO substrate. Also, we have the problem of highly variable activity of aldehyde oxidase in different lots of human liver cytosol or S9. And because of this, even for pooled products, the scale-up from human liver cytosol generally underpredicts the in vivo clearance by aldehyde oxidase. So in this paper from Pfizer, Zientek, and others, they report an average 11-fold underprediction of clearance. So this is in contrast to the generally correct in vitro to in vivo extrapolation for CYP-mediated clearance from microsomes or hepatocytes, even from the same lots. So I want to go into the variability that we see and then discuss some of the possible reasons before we move on. The variability, we reported this in 2007 in a poster at ISSX. We showed that across 24 individuals, we see a 20-fold variation with both phthalazine and vanillin. And we also looked at mRNA by RT-qPCR, and we see a 7-fold variation there, but there's a very poor correlation with activity, the R-squared value being less than 0.2. And then I want to mention two recent papers that have come out by Barr and then also from Fu and others at Pfizer. And in the Pfizer paper, they see anywhere from a 19 to a 90-fold variability in the intrinsic clearance from phthalazine all the way up to carbazeran across 20 individuals. And this is 50-50 male and female. And then another paper, they're looking at 3 different lots of pooled human liver cytosol from the same vendor, and they still see a 1.5-fold range in the Vmax for the aldehyde oxidase substrate DACA oxidation. So the variability in cytosolic AO activity across vendors, much has been made of this in the past, I believe, and we looked at this in our own experiments side by side, and we found that our flagship pooled human liver cytosolic product compared with 3 different competitors. You can see that with thalazine, we actually have higher activity than some of the others, but then for vanillin, we're lower than a couple of them. So within the same experiment, even vendor comparisons can be substrate dependent, but all of these provide rates within approximately 2-fold of one another. So the question is, what contributes to these variabilities that we're seeing? There's also variability in content. So this is 2 papers that have been published very recently, as recently as September 5th, that look at the content of aldehyde oxidase, and they do this by a mass spec method. So for the Pfizer paper, they're seeing a content of anywhere from around 1 to 2.3 picomoles per milligram protein, and a 3.4-fold variation, I should mention. So for the Barr paper, they see about a 2-fold variation by mass spec across 3 lots, again, of pooled human liver cytosol from the same vendor. And they see an absolute content of 21 to 40 picomoles on a per milligram basis. So if you look at that per milligram basis, there's anywhere from a 17 to 28-fold difference in the reported AO concentration. But these were 2 different vendors that were being compared, so that may contribute to the difference. There may be other factors as well. But the correlation, although not reported directly by the Pfizer paper, they can say that about 80% of samples, the aldehyde oxidase concentration fell between about 1 and 2 picomole per milligram protein, whereas the median is about 1.45. So again, correlation between AO activity and expression is weak. We showed this to some extent with mRNA. The Pfizer paper, they see little correlation. They show this in a nice graph where you can see the aldehyde oxidase concentration of the enzyme across samples. And then looking at carbazeran intrinsic clearance, what is of particular note are these very low activity samples have essentially normal aldehyde oxidase content. And then on the other end of the spectrum, you have samples that have very high intrinsic clearance for carbazeran. The Barr paper, they actually did find a weak correlation between aldehyde oxidase activity and aldehyde oxidase content with an R-squared value of 0.48. But again, this is for 3 lots of pooled human liver cytosol. So typically you wouldn't expect to see even that much variability across a pooled products from the same vendor. So the possible reasons for poor correlation between activity and expression. So individual factors have been put forward in the past. So for instance, age, disease state, smoking, and alcohol use. I've highlighted that because I'll touch on that a little bit later. But apparently not gender. I mean, we look in the Pfizer paper, they describe a 50-50 mix of male and female. Stability, much has been made of this, so especially warm ischemia time from when the liver is collected to actually making the fraction makes a big difference. Sample preparation, handling, and storage, as well as buffer components. And then the properties of the enzyme. It's a complex enzyme. It forms a dimer, so you can have homodimer dissociation, protein misfolding, even chemical or enzymatic modification have been put forward as possible reasons for this poor correlation. You also have variable correlation, incorporation or desulfurization of the molybdenum cofactor. And of note is the fact that these various inactive or less active forms would not be differentiated by these mass spec methods. And then genetic variability. So Hartman and others have described in the last year poor or even fast metabolizers, looking at an Italian cohort of about 180 individuals and looking at different polymorphisms. And then there's also the idea that there may be translational regulation, and that maybe even adiponectin levels could influence that. What about chemical inhibition? We have looked at this in the past for some other subcellular fractions, and whether or not a small amount of a drug that one of the donors may have been taking. So, raloxifene is a very potent inhibitor of aldehyde oxidase. But then alcohol use, this is a very interesting and intriguing possibility from the Pfizer paper, where 2 individual donors with the very lowest activity but normal aldehyde oxidase concentration were heavy chronic alcohol users. And a quick look at our data from 2007 revealed that 5 of the lowest activity donors, which had less than 33% of average, were also alcohol users. And then what about the freeze-thaw long-term storage stability, so other handling issues that people have mentioned? So we've looked at this, and we will be presenting this next week in Toronto at the ISSX meeting. We looked at our human liver S9 pool from 2004 through 2011, and whenever you pull these out over time, there's essentially no trend. You get essentially the same activity over time. So freeze-thaw or long-term stability is not a problem. Even freeze-thaw cycles do not appear to be a problem. You can see that for our first freeze-thaw, that actually had the lowest, and then all the way up through cycle 10, we don't see a decrease. We certainly don't see a trend in that. So there's no impact of multiple freeze-thaw cycles or long-term storage. So what about the impact of fresh versus frozen tissue on aldehyde oxidase activity? So we actually had a few analysts that as fresh livers came in, would then process that tissue immediately into S9 and determine activity with thalazine, or they would take the fresh tissue, freeze away the S9, or then take the frozen tissue, immediately prepare S9 and look at the activity, or the fourth variable, take the frozen tissue, take the frozen S9 the way it typically is done, and look at the activity across 3 different livers. And you'll see you certainly don't see a trend for the frozen to be lower. And across all these individuals, the activities are within 50% of one another, regardless of the use of fresh or frozen tissue, or whether you use fresh or frozen and then thawed S9. So even this sample handling aspect does not appear to impact the aldehyde oxidase activity. So what about the impact of chemical inhibition? We looked at dialysis. And just to show you the way this experiment is designed, we have dialysis for 4 hours. So our control for this is the refrigerated sample for 4 hours, and then we compared that with freshly thawed S9. And you'll see that there's really no trend. In fact, dialysis sample was actually lower than the others, which would not be consistent with chemical inhibition. So there's no apparent endogenous or contaminating inhibitor present by this experimental design. So just to summarize the possible reasons for variability, obviously you have individual and possibly even environmental factors, the stability of the aldehyde oxidase, the particular issues associated with the enzyme itself, as I mentioned, the molybdenum cofactor incorporation, and then genetic variability. A number of single nucleotide polymorphisms have been described, and then other mechanisms that we don't understand yet. So there's a lot of work yet to be done here. Very interesting, but I think Faraz will go into details of why most of these things, when you're looking at subcellular fractions, don't necessarily matter as far as the impact of either low or high activity and the extrapolation of clearance for drugs metabolized by aldehyde oxidase.
Thanks, Brian. So I'll get right into this. Traditionally, for P450s at least, the allometric scaling approach using in vitro intrinsic clearance and scaling that to the in vivo clearance values is well established, but that paradigm is not necessarily in place for aldehyde oxidase. And as a result, the in vitro to in vivo extrapolation is really challenging, in large part due to the chronic underprediction by the in vitro test systems in predicting the in vivo clearance values. And Brian alluded to that a little bit in his earlier slides. And this may be why in the industry high activity, quote-unquote high activity aldehyde oxidase subcellular fractions are generally preferred, because that may influence the underprediction and actually minimize that underprediction if your in vitro activity is higher. That being said, a couple of years ago, a paper out of Pfizer was really the first report to do an IV-IVE approach with aldehyde oxidase using in vitro metabolism data for aldehyde oxidase substrates that had corresponding in vivo PK data available as well. So this was a paper by Mike Zientek with Scott Obuck as a senior author. And they described a yardstick or rank order approach to essentially benchmark a new chemical entity with respect to existing aldehyde oxidase substrates in order to gauge the level of clearance for a particular new chemical entity. And I'll get into a little bit about how they did this approach. So they examined 11 drugs that are primarily metabolized by aldehyde oxidase and that had corresponding in vivo PK data. And they determined the in vitro intrinsic clearance values with pooled human liver cytosol, or S9, and compared that back to the free in vivo intrinsic clearance that was observed with the in vivo data. And they have a penultimate figure that summarizes all of that in their paper. And essentially, they've sorted their data such that those substrates that are in vivo high clearance aldehyde oxidase substrates are on the right-hand side. We've got a group in the middle that comprises the medium clearance aldehyde oxidase substrates, and then the low clearance aldehyde oxidase substrates. You can see that, just like Brian mentioned in his earlier slides, that some of these classical AO substrates fit into these molds. Carbazeran is a high intrinsic clearance substrate in vivo, and we see that partly reflected in the in vivo as the highest in vitro intrinsic scaled clearance value. We've got zaleplon, which is in the middle, and we've got drugs like zaleplon and methotrexate, which are on the low end. Essentially, what this paper suggests is that this correlation allows for qualitative scaling for a new drug, and that a new drug can be categorized as a low or medium or high clearance substrate of AO by running several of these drugs as calibrators alongside a new chemical entity. So for example, if you were to take zaleplon, zaniparide, and carbazeran and run that alongside with your drug of interest in either S9 or cytosol, you can essentially rank order your new drug relative to those other known AO substrates and get a feel for whether your drug is going to be in the middle and towards the high end or towards the low end of the in vivo predicted in vivo clearance. So the question we pose here at XenoTech is that is this rank order approach used to benchmark these new chemical entities advocated by the Pfizer paper, is it really dependent on the absolute AO activity in S9 or cytosol? Meaning that do you absolutely have to have high AO activities in these subcellular fractions in order to do this approach? Just before I get into the data, all of the data that I'll be summarizing in the next couple of slides we'll be talking about is going to be presented next week at ISSX as well. Poster 331, so if you're attending, be sure to stop by there and pick up a copy. But diving straight into what we did, so we looked at the test set compounds that the Pfizer paper used with diverse intrinsic clearances for AO, and selected those compounds that had high, medium, or low in vitro intrinsic clearances, and used this rank order approach with multiple human individual lots. In this particular case, we used 5 human individual lots of both S9 and cytosol, and we also examined our pooled products here. We looked at our pool of 50 cytosol product and our pool of 200 S9 product. And it's important to note that these 5 individuals, including our pooled products, span almost a 10-fold difference in AO activity, which we determined by thalazine. So our premise was that we would look at these test set of compounds, determine the half-lives of these AO substrates in all of these subcellular fractions, from the individual lots as well as the pooled products, and use the rank order approach and see, are we able to get the appropriate rank order? Do high clearance drugs have a higher clearance? Do low clearance drugs have a lower clearance? And are we able to establish that benchmark? So diving straight into the data, This slide depicts our initial data set with human cytosol. So the top three graphs here depict, like Brian mentioned earlier, the classic substrates that have high AO intrinsic clearance, medium and low intrinsic clearance, carbazeran, zanipride, and zaleplon, respectively. You can see that we've got the 5 individuals plotted here, as well as our pooled product, and as expected, and this is in order of increasing activity as well. So our H1 has the lowest activity, our H5 has the highest activity. So as you increase the activity, by definition, you are increasing the clearance of these drugs, which makes sense. Diving deeper into the data, when we use the rank order approach, and again, we've got high clearance AO substrates at the top, we've got the medium substrates grouped in the middle, and we've got zaleplon and methotrexate, which are the low substrates at the bottom, and a marker of AO activity, phthalazine. And the individuals rank ordered from left to right in terms of their AO activity. We've got 382 as the lowest activity individual in H1, all the way up to H5, which has the highest activity individual. So we're spanning about tenfold, and our pool is right about in the middle of the activity. You can see that, for the most part, the rank order approach worked in every case, with the exception of the lowest activity individual. And this suggests that we need to establish, at least for human cytosol, potentially a threshold value, or there is a threshold value after which this rank order approach in cytosol is appropriate. You can see here that in a particular individual, for example, if you were to take H2, that the half-lives for carbazeran are very short. They're slightly longer for the medium clearance drugs, and then they're really long for the low clearance drugs, consistent with high, medium, and low clearance, and we're getting the appropriate rank order within an individual. That persists throughout H3, H4, H5, as well as our pooled product. That is not the case for our lowest activity individual, where only carbazeran we're able to achieve a half-life, and then we get essentially no detectable loss in the duration of the experiment for the other drugs. So again, like I mentioned, this suggests that a threshold value is needed, at least for human cytosol, to establish the rank order approach. And based on our data here, our threshold value would be right in between human 1 and human 2. That beyond human 2, we're able to establish the rank order approach. And just to again highlight that our pooled product, it reflects the median activity in the span of AO activities that we examined. Looking at human S9, again, we've got substrates that have high clearance, medium clearance, and low clearance, like the previous slide. And then diving straight into the actual half-life values, you can see that the rank order approach in the case of cytosol, or in the case of S9, actually worked in every case, including the lowest activity individual. And we see here that in every individual, H1 through H5, again, going from lowest activity to highest activity, we're able to achieve the appropriate rank order that carbazeran is the fastest, followed by the medium clearance drugs, followed by the low clearance drugs. So again, this suggests that for S9, that our lowest activity individual is above any potential threshold value and that would be needed to determine this benchmark rank order approach. Again, in terms of S9, which is our pool of 200 product, the AO activity reflected the median activity in the span of AO activities that we examined. So in answer to the question that we posed earlier, is this rank order approach that this Pfizer paper published on, is it dependent on the absolute AO activity in the subcellular fractions? Well, the answer is it's not dependent on the absolute AO activity. And we showed that with the cytosol and S9, that even with low activity individuals, we're able to correctly establish the rank order for the calibrator compounds in the test set, enabling you to determine where your new drug would stand relative to those other drugs, with the exception of cytosol, where the lowest activity individual, we were not able to achieve that. So there may be the need to define certain thresholds for the subcellular fractions to demonstrate that the rank order approach is appropriate with that particular product. To determine the threshold activity, we would recommend doing a characterization of the subcellular fractions with the test set of compounds like the previous slides, to determine whether a particular product is appropriate for the rank order approach. If you're able to get the high clearance drugs as the fastest, and the medium in the middle, and the low on the long half-lives, then that basically you can be assured that you can get a benchmark for a particular new drug. It's important to note that XenoTech has done this for our pooled products, again for our pooled S9 and our pooled cytosol, that we have established that these products are appropriate to use for the yardstick or rank order approach. It's also important to note that pooled products by their nature will intrinsically have AO activity above a hypothetical threshold, assuming if you have enough individuals in a particular lot. And that's been demonstrated in a couple of literature sources as well, and we show that here in our data as well, that our pooled products reflect about the middle of the pack activity in terms of the span of activity that we see with AO. Getting into some other aspects, I'll talk a little bit about how to calculate whether your drug is metabolized by AO. So traditionally, this approach, the approach used for CYP enzymes at least, is the use of chemical inhibitors, and this is widely used in the industry with various subcellular fractions or intact systems like hepatocytes. You can use microsomes, S9, or hepatocytes, and essentially you can use a specific chemical inhibitor to knock out either all P450 or specific isoform inhibitors to determine the contribution of particular P450. So for example, in the case of if you wanted to know whether your compound is metabolized by CYP3A4, you could use a CYP3A4 specific inhibitor. If you just wanted a general idea for general P450 contribution, you could use a PEN specific inhibitor, such as 1-aminobenzotriazole, to knock out all your P450s quite effectively. A similar approach, of course, can be used to determine the fractional metabolism by AO using a AO-specific inhibitor. And recently, last year, another Pfizer paper from Scott Obach's group published on using hydralazine as a specific selective probe inactivator for AO in human hepatocytes. So we went ahead and took a look at whether hydralazine would be a good candidate to determine FMAO like they did in the Pfizer paper. Just before I dive into our data, just to orient you on a table that was in this paper, you can clearly see that for many drugs they were able to determine the FMAO using this hydralazine approach. And we went ahead and investigated whether we could use 1-ABT and hydralazine and mimic the results in the Pfizer paper. So getting right into what we obtained, here's a set of compounds that we examined with varying metabolic pathways. We looked at the intrinsic clearance of these compounds in pooled human hepatocytes using our flagship HepatoSure product, which is a pool of 100 individuals' hepatocytes. And we dosed these hepatocytes with an inhibitory concentration of hydralazine. So with 25 micromolar hydralazine, you would, in theory, knock out all of the AO. And we also did treatments with 1 millimolar aminobenzotriazole, which you would knock out all the P450. And you can see here that for substrates that are, we know that are metabolized by AO, such as carbazeran, benzylguanine, zaleplon, zaniparide, we actually confirm contribution from AO towards the metabolism in that once we dosed with hydralazine, the intrinsic clearance actually went down quite a bit. And we see that reflected here as a FM value. Interestingly enough, we also saw a contribution for AO with CYP2D6 substrates, and these are substrates with high FM, at least dextromethorphan is a high FM CYP2D6 metabolic contribution. And this was unusual to us because, to our knowledge, these substrates are not necessarily metabolized to this degree by AO. And Scott Obach's group and the Pfizer paper saw a little bit of this as well. And it turns out that hydralazine is actually a CYP2D6 inhibitor in hepatocytes. And The Pfizer paper looked at this, and they saw a little bit less inhibition than we did for hydralazine. We get an IC50 of about 18 micromolar for CYP2D6, and it seems to be, at least in the panel of CYPs that we examined, quite specific to CYP2D6. So our conclusion is that hydralazine may be useful to do this chemical inhibition type of assay in order to determine the fractional metabolism of a new drug by AO, but only if you're confident that your drug is not a substrate of CYP2D6 as well. So if your drug is not metabolized by CYP2D6, go ahead and use hydralazine to determine the AO contribution if you suspect AO is involved in its metabolism. If your drug is also a substrate for CYP2D6, some additional controls will probably be necessary in order to tease out exactly the contribution for AO, such as using a specific 2D6 inhibitor and an additional CYP2D6 substrate. So just before I move on to the next slide, it's also important to note that the chemical inhibition approach here described here for hepatocytes can also be used with S9. So you can dose hydralazine in S9 and get a similar type of representation of data as well. All right, so I'll just go through some concluding remarks that we went through today. So Brian talked a little bit about the importance of AO as more drugs are moving away from P450 metabolism, and we're designing drugs in the industry to move away from phase 1 metabolism. More of these non-P450 enzymes are coming into play and into greater importance, such as AO. And Brian mentioned some of the structural alerts associated with substrates of AO as we move away from P450 metabolism. He also discussed the variability in AO activity, and that it doesn't necessarily correlate with the content of the enzyme in the particular subcellular fraction, or it doesn't even necessarily correlate with the expression of that enzyme as well. So there's high variability in these subcellular fractions, and content doesn't necessarily give you a good picture of what your activity would be. The sample prep and various handling and procedural processes do not appear to explain this difference in variability. There is an intriguing possibility, like Brian mentioned, that some environmental factors like alcohol use may play a role, but again, there's not a lot of data out there yet to make a concrete correlation with that. I talked a little bit about the rank order approach that the Zientek paper used to qualitatively benchmark the in vivo intrinsic clearance for AO. By using these calibrator compounds, you can get a feel for where your compound will fall with respect to those compounds. It's important to note that this approach is not dependent on the absolute AO activity of cytosol or S9. You don't need high AO activity lots to do this approach. You can, as long as a particular threshold has been established for a subcellular product, you can use that product for that rank order approach. Again, characterization of those products is important to determine whether those thresholds are met. And again, XenoTech's products, our pooled S9 and cytosol products, meet or exceed that threshold and are suitable for use with the rank order approach. Lastly, I talked a little bit about inhibition of aldehyde oxidase by hydralazine as a tool to specifically determine the fractional metabolism of AO by a new drug. It's a nice way to determine the FMAO, much like 1-ABT or specific inhibitors are used for P450. You can use hydralazine to determine the AO contribution, but it's important to note the effects of hydralazine on CYP2D6, namely the inhibitory effects, and I would caution use for hydralazine as a tool to determine FMAO if your drug is known to be metabolized by CYP2D6. There may be some additional controls that may be needed to further elucidate that effect. With that, we'd like to thank you for attending our presentation, and we'll be happy to take any questions that you may have. We've got a bit of time to answer that, so I'll pass it over to Matt, and then we'll entertain your questions.
Thanks, Faraz and Brian. As a reminder, if you guys have any questions regarding the presentation, please remember to type out your questions in the questions pane on the GoToWebinar user console. Before we move into Q&A, I would like to share with you all a few upcoming events where XenoTech will be participating. Next week we'll be at the 10th International ISSX meeting in Toronto. Our booth number is 301, and we are presenting 11 posters at this year's event. Our scientists have done a tremendous amount of work, and if you would like to see a list of those posters, you can visit our website, and it's on the ISSX event column. On the bottom right-hand side, you'll see the posters listed. Also, if you'll be in attendance, please feel free to stop by the booth and request a reprint of any of the posters or talk with our scientists directly at their poster presentations. Also, on October 17th, we'll be at the North Jersey DMDG Symposium. Our Director of Analytical Services, Dr. Joanna Barbara, will be presenting Exploring the Mechanism of Metabolic Intermediate Complex-Based Metabolism-Dependent CYP Inhibition, the case of lapatinib. So if anyone will be in North Jersey DMDG, please stop by and talk with her. All right, I'm going to move it over to questions and answers. I don't know if we had any questions. Let's see. Did we have any questions, guys?
Yeah, so there's a couple here about using substrates in mice strains for aldehyde oxidase, and that some strains of mice do have high aldehyde oxidase activity. So I would say, and I would caution you to remember that mice have multiple forms of aldehyde oxidase, and there are likely differences in the enzyme between the single human aldehyde oxidase and the multiple forms that are present, two major forms in mouse liver in particular. So while it certainly may be possible, I don't know that there would be a good concordance, and some of the recent work that has come out, even looking at guinea pig and rhesus monkey, which appear to be closer to the human situation, there is still a substrate-dependent effect. I mean, we see a substrate-dependent effect even within the single aldehyde oxidase in human. So that would be my word of caution there. Sorry, so the next question, how stable is the AO activity for long in vitro incubations, S9 and cytosol, particularly when incubations conducted for longer than an hour, the AO activity comparable at 4 hours relative to when the assay begins? We've run these out to 240 minutes and observed linearity, and I do remember at least one paper, I apologize, I can't recall offhand who it was that published it, where they were able to see linearity of these low clearance AO substrates for around 780 minutes, if I remember correctly. So I think it probably depends on the substrate as to whether or not you see linearity, and the stability may be an issue depending on the buffer composition and so on.
So the next question is, do you get similar clearance in hepatocytes with the panel of compounds you use in cytosol and S9? So that's a great question. So if we were to use a test set of compounds, we have not done that type of experiment in depth. We know that there's a nice paper from Matt Hutzler, I believe 2012 in DMD, where they take a similar approach to what the Zientek paper did, and try to determine whether using cryopreserved hepatocytes is appropriate to do scaling for AO. And they see, for some substrates, they see a modest improvement, but again, there's still an underprediction with hepatocytes. There's no real simple way to essentially improve the scaling and get out of that underprediction, essentially.
Okay, I can take.
Okay, go ahead.
Sorry, I was going to say there's a couple of questions here about raloxifene as a chemical inhibitor. And yes, I mean, I think raloxifene was reported as the most potent direct inhibitor of aldehyde oxidase. I think the shift to hydralazine has largely to do with the fact that it's a mechanism-based inhibitor of aldehyde oxidase, so you don't have to worry as much about the time course of the inhibition if you completely knock it out. Also, the raloxifene will inhibit other CYPs, if I remember correctly, even if you are using a relatively low concentration. But I think that has been, in the past, algorithms that you see published. You know, if you want to differentiate between P450 and aldehyde oxidase, use raloxifene. But I think given the newer data with hydralazine, that would be our preference, with the caveat that Faraz mentioned about CYP2D6 inhibition.
All right, so I'm gonna jump up to this question. Do you think it is better to use S9 for metabolic stability screening or to use microsomes? And that's an interesting question, and really it depends on what type of question are you trying to answer. If you know, or if you're concerned with just P450 stability, and not necessarily concerned, or if based on the structural motif of your drug, you know that it's not necessarily going to go through AO, some of these other enzymes, and that if you're concerned with primarily just P450 and UGT, for instance, then doing stability screening with microsomes may be appropriate. If, for example, you do this type of assay in hepatocytes, and Brian alluded to this, that if you see higher clearance in hepatocytes relative to a simpler test system like microsomes, then perhaps then it's appropriate to go back and look at maybe S9 and see is there a contribution from a cytosolic enzyme, and tease out whether there's an AO component as well. So really, it just depends on the type of question that you're answering. There's no real straightforward answer whether it's better to use S9 or microsomes. It really depends on the indication and the program that you have set up. Yeah, we answered that question to this question.
Sorry, we're just sorting through some of the questions here. So explanation of why the AO activity was in pooled lots was reported as median and not mean. And I think that was just in a couple of presentations, or sorry, publications that it was mentioned that way. Faraz mentioned that in terms of the median activity, and that may be because of the non-normal distribution that we see across some samples.
Have you, okay, so I'm going through these questions. Have you seen concentration differences in kinetics as you increase and decrease of your probe substrates? I don't know if we see differences in kinetics off the top of my head. The only thing I could think of is that there are substrate-specific differences that we see, and Brian alluded a little bit to that, but I'm not sure if I'm understanding the question correctly.
And also, there are some substrate inhibition or possibly product inhibition that occurs with some substrates. I actually wondered if that difference in kinetics is referring to inhibition kinetics. And so some inhibitors appear to be uncompetitive inhibitors of aldehyde oxidase versus competitive. There's a question here about using hydralazine as an aldehyde oxidase inhibitor relative to xanthine oxidase, and that I don't know for a fact has been examined. I will say that xanthine oxidoreductase tends to have much more selective substrate specificity than aldehyde oxidase. So there are, of course, some drugs that are metabolized by xanthine oxidoreductase, but I think there are far more drugs that are metabolized by aldehyde oxidase. So I'm not sure that would be of great importance, but certainly could be validated and maybe an important thing for someone to do at some point. Okay, we're looking at a follow-up question that says, sorry, if we run the clearance assays using 1 versus 100 micromolar phthalazine, and I don't remember offhand if we did that in particular. We're probably looking at a concentration that allows us to determine clearance easily, and I don't know offhand if we have those data.
All right, Brian, I think I'm going to, unless you guys have any other questions that you're wanting to answer before I close this off. Any other questions?
No, thank you all for the great questions. We appreciate it.
Yeah, absolutely.
All right, guys, I'm going to go ahead and wrap up today's session. Thank you all for taking your time out of your day to join us, and a special thank you to Brian and Faraz for their presentation and answering those questions for you guys. Up on the screen, you'll find our general contact information. You can always contact us if you have any questions regarding today's presentation or products in general, S9, cytosol, give us a phone call, 913-438-7450, or email us at info@XenoTechLLC.com. Thank you for joining us today, and have a great rest of your day.